Okay, well, it's great to be here today, and we'd like to share with you a case study illustrating CDMO selection for bispecific antibody development, and in particular, the need to match capabilities of CDMOs to the needs of the sponsor. Just to give you an idea of what we're going to talk about for the next hour or so, I'll first introduce the challenge, and that is the client company and the project, a target product profile, and then how to use a consultant in this process. Then we'll talk about the match, that is considerations for candidate CDMOs, switch to the scope of work to dig in a little bit to the development stages of work, GMP production of clinical trial material, and also the timing of regulatory interactions, specifically a pre-IND meeting, and then the IND itself. And then wrap up with a summary. We did provide you in advance with pre-reads. You may have seen these. These are a few articles that we thought would introduce you to the topic and give you some good background. The first is an article by Judy Myers that was published about a year ago in Pharma Manufacturing, How to Find the Right CDMO Partner in Pharma. The second is an article by Stephen Kloss that was published a few years back in Chemistry Today about QBD and working with your contract manufacturer. And then the third one, an article by David Wilson that appears on the ZymeWire blog earlier this year. Article about outsourcing in pharma and biotech, how to use contract manufacturers. So these are a sampling of articles that you might want to read. If you haven't already, then the links to these articles are on our website. So I want to talk about the challenge that is going to be the topic of today. So the client here is a small venture-funded Bay Area biotechnology company. This is a common type of profile, I guess, of a company in our portfolio. They have internal capabilities. They have a proof of concept research laboratory, but no CMC development infrastructure. And we see this a lot, companies that are venture funded, that have discovery technology, prefer to focus their efforts and resources on potentially generating new products, new antibody candidates, and prefer not to invest the considerable resources in the development infrastructure that may be necessary, that will be necessary. And so often these kinds of companies look to outsource these functions, and that's where consulting groups like ours come in. So this particular client has a monoclonal antibody discovery platform and an oncology focus. So the project that this sponsor is working on is a preclinical monoclonal antibody. Now, the topic, as we have indicated, is bispecific development. But in fact, what we would like to do is talk to you about this humanized IgG1 project. It is not a bispecific, but the development program is very similar. The challenges that one faces, the tasks that are needed to do for a full-length IgG and a bispecific are similar. We will point out during the course of the talk where bispecifics differ from the development of a monoclonal antibody, and talk specifically about some of the issues that we have seen and that you're likely to face with a bispecific that makes them unique. This client company hired an external consulting group to execute CMC. Our role was to identify, evaluate, select, and manage a CDMO, to develop a pre-IND strategy, and to write the CMC module of the IND. Our starting point was the in silico amino acid sequence and a target product profile. This is basically, we talk about the product development decision that sponsor companies make, which is the transition from research to development. And the in silico amino acid sequence and the TPP are both happening as a result of the product development decision. So what is the target product profile, or the TPP? This is a very important document that we work with sponsors to develop. It's basically a needs checklist. And what it does is it defines the product's characteristics. So it would be a product description, what are the clinical indications and usage, the dosage and administration, dosage forms and strengths, and how the drug is supplied and handled. This document, a TPP, provides the client with a roadmap in order to guide product development. So for example, the anticipated dose and the clinical indications defines the amount needed for clinical studies, which then defines the scale of production. And so since we need to know the scale of production in selecting appropriate CDMOs, the TPP gives us the linkage between the anticipated dose and clinical indications and what the scale of production needs to be. Also, the route of administration defines the type of formulation. And so that's another thing that we need to have visibility on at the beginning of the program. So this is an example of a preclinical TPP that's partly completed for this program. So you'll notice that the clinical indication is oncology. The scale of production is 2,000 liters or potentially 1,000 liters. The route of administration is infusion, and so it suggests a 10 mg per mil liquid solution and 2 to 8 degree storage. So if you look at the TPP, you see that in the left column are characteristics and the right column is the detail. We have here the molecular description as a humanized IgG1 kappa, the drug substance stability, 24 months at minus 20 degrees C. The dosage form is a buffered liquid solution. So this a TPP like this can be completed without knowing a lot about the product. And it's a living document, so that we revise the TPP as we learn more about the product itself. And what it does is it gives all of the stakeholders involved in the program, the sponsor, the consultants, the vendors, everybody understands what it is we're trying to accomplish with this drug product and drug substance. And so it makes it much easier to be working toward the same goal. All right, so with that, then as the introduction, I would like to tell you a little bit about the match. And to do this, I want to introduce Tracy Konjerski.
Thank you, Steve. Okay, so over the next few slides, I am going to review the outsourcing model where a client needs cGMP material and they choose to utilize CMC consultants in their process of selecting and managing their CDMO partners. So on this slide, this really shows the overall model that I'm talking about. As I mentioned, we have a client looking for GMP material. They use a CMC consultant because we provide industry expertise. And in the end, we ultimately want to be sure that the client is getting the GMP material that they need to take forward for their clinical trials. So CMC consultants are industry experts. We provide technical, quality, regulatory, as well as business and negotiation expertise to the client throughout the entire process of evaluating and selecting their CDMO. We also help them manage the CDMO relationship as they go forward. And we really function on behalf of the client. We function as an advocate on behalf of the client, especially where um, interactions are involved with the CDMO. And if things get challenging, we are the ones that sit on the other side of the table and work through those challenges and those situations to help ensure that the client actually gets to move forward to the successful end of their project. Um, Now, the CMC consultant is really a good support or a bridge for clients who have very little experience in outsourcing and little experience working with CDMOs in the industry. So by using a CMC consultant to help select and manage that process of selecting the CDMO, the outcome is typically more positive for those organizations that need the help that we can bring to the table. Next slide. Okay. At the start of the CDMO selection process, the CMC consultant provides the client with a list of CDMOs to be considered for their project. Now, all of the CDMOs that are included in the evaluation process have experience that's directly related or relevant to the client's molecule, except they vary between their specific amongst the CDMOs. The expertise varies as well as their capabilities vary. So while we know, for example, every CDMO would be included in the evaluation of a cell culture based early phase development and manufacturing project, we need to help the client understand how these CDMOs are differentiated in the various molecules that they've worked with in the past, what their track record is, how their success in working with other clients relates specifically to their molecule. And we really look for the CDMO to provide solution-based results and examples of their experience to us. So there are some CDMOs that focus solely on monoclonal antibodies, while others have developed a strong track record in the industry in working with antibodies, which are more bispecifics or trispecifics, or just basically more complex antibodies than a standard mAb. We have other CDMOs who are known for focusing on enzymes or blood products, such as AGC, for example. Other CDMOs are very well versed in working with ADCs and helping the client manage the ADCs have a more complex supply chain and approach to going from start to completion. And so CDMOs such as Piramal is an example of one organization that can help clients when they are taking ADCs forward. In addition to looking at the details of the expertise of the CDMOs, we also look at the capabilities. And there are a number of factors that we can consider when looking at this topic, but I'll just mention a few here. One of them that we evaluate is whether or not a CDMO is a one-stop shop, or if they focus more on one or two core competencies. We also look at the CGMP manufacturing facilities that the CDMO has and their overall operations. We evaluate the CDMO based on whether or not they have a gene to manufacture platform that helps to simplify the process for the clients, such as the IBEX platform that Lonza offers. So we look at a number of different things when it comes to expertise and capabilities. And of course, in terms of time, we're not mentioning everything here today, but those are just a few considerations. Next slide. Okay, so in addition to evaluating CDMOs based on their expertise and understanding their capabilities, also important in the process of selecting a CDMO is the ranking of the factors that are important to the client and how those factors align with the CDMOs that are evaluated in the process. And again, on the next couple of slides, I'll talk about a number of factors that show up over and over again, but this is not by any means an exhaustive list of factors that may come into play. Those indicated in red on this slide were the factors that were prioritized by the client who is the subject of today's case study discussion. Another thing to note is that, again, even if the same factors show up over and over, when we talk to different clients will rank those factors differently. And that is what impacts the difference in the outcome in the selection process and which CDMO they decide to work with. So very important is ensuring that we match the molecule's phase of development to the actual experience and capabilities of the CDMO. We want to make sure, for example, if our client has a pre-IND molecule, that we are pairing them up with a CDMO that has relevant experience, knows how to organize their resources, their infrastructure, and the approach to that project. And this is very different from the approach to, say, a late phase or commercial molecule. Another set of priorities that and factors that show up very often when we are working with clients, and these pretty much come together: quality, cost, and time. Quality is a given, and at a minimum has to be phase appropriate and is non-negotiable, whereas cost and time are typically considered the two trade-offs, depending on what the client has more of, whether or not they have more money or whether or not they have more time. So the client's project drivers will help to prioritize their factors for them here. Another consideration for very early phase development and manufacturing projects is the cell line that the CDMO offers, as well as their experience and capabilities in developing that from the cell line to develop the process moving forward for to take to manufacture. So in terms of evaluating the cell line, clients often go into the selection and evaluation process thinking they may want a proprietary system, some want a non-proprietary system. And it's really important for clients to understand the pros and cons of working with a particular system, knowing what productivity they can expect, understanding what the terms of access are to those to the particular system, how it impacts them early on, how it impacts them financially, downstream, when the molecules move through to the later phases of clinical trials. So it's important that we give the client a view to what it means to work with a particular CDMO cell line and compare that across all of the CDMOs included in the evaluation process. And further, it's important for the client to really have a view into the experience that the client has, or that the CDMO has, in developing the cell line, and from the cell line, the cloning, and selecting the actual cell line that's going to move forward into the process development. We need to make sure that they're good at providing them with a line that is robust and scalable, is going to have good stability, going to have the productivity that they're expecting. And so these are all pieces of information that we're gathering for the client along the way in their process. Next slide. Again, we look at whether or not a CDMO is a one-stop shop. Some clients are looking for that, others are not, and it really depends in the end, the information that they gather and what's important to them. We are looking for CDMOs who can successfully perform the process development for early phase projects, their formulation development strengths, what their analytical methods and testing and stability packages look like, typically in terms of timeline and cost, their experience and capabilities for manufacturing the cell lines, characterizing these. And again, we look at the GMP manufacturing, the scales of manufacture, the systems or the platforms that they deploy for the manufacturing, and things related to all things considered around GMP manufacture. So this, again, is not a full list of factors that clients consider. It's just a place to start for today's discussion. Next slide. Okay, and on this slide, the color-coded figure that you see here is representative of a very early stage process development and manufacturing project. The the figure actually illustrates the three primary stages in this type of project and the tasks on high level that one might expect to have going on in each of these stages. And we've also indicated what the typical timeline is for those three different stages. Overall, the duration of a project similar to this, from amino acid sequence to generation of cGMP material, packaging, labeling, and readiness for for the clinical trials, shouldn't take more than 15 to 20 months. Some CDMOs are able to accomplish this in a shorter amount of time, but it shouldn't have to go over 20 months, 21 months. And this also is demonstrating how an early phase project like the one we're talking about in this case study today can end up involving 3 separate CDMOs in order to take the client from start to completion. And so this project starts with the amino acid sequence being delivered to the CDMO1. CDMO1 initially right away will start cell line development, complete process development, generate tox material for the clients to use, and then move on to producing the final GMP drug substance material. The drug substance material transfers to CDMO2, where the drug product activities take place, and the GMP drug product is then transferred to CDMO3 in this scenario for packaging, labeling, and distribution for use in clinical trials. Now, again, we show how this project could break out and be distributed amongst 3 different CDMOs for one client, but another client, based on what's important to them in their process, may decide to work with a one-stop shop that could take them all the way through from start to completion. And in this particular case study related to today's discussion, the client for their early phase project ended up choosing 2 CDMOs for their project from start to completion. So just in closing, I just want to mention that in partnering with CDMO consultants, the client benefits by being able to take advantage of the experience and the guidance that the CMC consultant brings to them. And they can apply this is applied throughout the CDMO selection process. And this will allow the client to make a very well-informed decision and help them to choose a CDMO or CDMO partners that are best aligned with their project requirements. And so with that, I will turn the presentation back over to Steve.
Thanks, Tracy. Okay, so let's switch to talk about the scope of work for this particular program. So the starting point, as we said, was synthesizing cDNA from any amino acid sequence. This is an example of a monoclonal antibody gamma-1 heavy chain amino acid sequence and cDNA sequence. In point, a deliverable as a starting point is the amino acid sequence to allow the CDMO to do codon optimization in designing and synthesizing the cDNA itself. So we prefer to provide that amino acid sequence as a starting point. And then the goal is producing bulk drug substance and filled and labeled drug product, which are shown here. So the bulk drug substance is typically frozen in bags, as you can see here. The drug product is vialed and labeled typically in a vial. So the development scope of work. that we undertook. So we started with cell line development, and you see on the right the graphic that Tracy had shown you with the development stage highlighted. So the cell line development starts with cDNA synthesis and plasmid construction. And cell line generation itself has as the endpoint to generate a research cell bank. This is a single cell cloned demonstrated monoclonal line that is stable and characterized appropriately. That is then used as a starting point for generating a master cell bank. We use the research cell bank to create a process for manufacture. That process will have as its components the upstream process, some data for which I'll show you in a minute, the downstream process, which is also the purification, and the formulation that will be used for both the drug substance and drug product. That's typically for monoclonal antibodies and antibody derivatives the same. The drug substance and the drug product formulation is typically the same. In terms of analytical, there are compendial methods as well as product-specific methods. The physicochemical methods are generally provided as the CDMO's platform methods that are adapted to the product of interest, with the exception of the potency method. That's going to be specific for each individual product, and that typically comes with the amino acid sequence from the sponsor. With the process developed, then the scale-up phase and the clinical production phases of work are undertaken. We start with tox production, where the pilot batch that is used to produce tox is also producing reference material. And we want to get preliminary drug product stability at this point. So often we will request of a CDMO that the tox lot go beyond drug substance, but also include a mock fill in appropriate container closures that will then be used for preliminary drug product stability. The reason that we like to do this is because it gives us an early window on drug product stability of the molecule. Eventually, we're going to file the IND with only 1 month of real-time drug product stability. And so if we have this preliminary drug product stability from several months earlier, then it gives the agencies a little more confidence that in the stability of the molecule. So that's why we like to include this activity as part of tox. We'll characterize the reference material, and then this reference material from the tox lot is used to release the GMP batch. From the GMP batch, which in this case is 2,000 liters, we took material for a viral clearance study, and we do ICH stability. We then went on to produce a GMP drug product and use this to do ICH stability as well. We expect that the CDMO understands that stability from both of these GMP batches needs to be ICH. Once in a while, not too often, but once in a while, we will get proposals from CDMOs that include stability study designs that are not compliant with ICH, and we really have to scratch our heads about that, but they need to be ICH at this point. And then that goes on to clinical labeling, packaging, and distribution. Okay, so a little bit about the data that we were able to accomplish in this particular program. The elements of the process, as you all know, we start with a cell line that goes to the upstream process, which is the expansion train through bioreactor production. That goes to the downstream process, purification of the product, and formulation and testing. So our first consideration is the cell line and the expression system. As Tracy pointed out, there are two types of cell lines that can be considered, or expression systems that are available. One is proprietary, the other is non-proprietary. And these differ in a variety of ways, as you can see on this table. So the proprietary type of expression system, it requires a license. And the expected productivity tends to be higher than for non-proprietary systems, potentially significantly higher. And it may also require proprietary media. The timeline also tends to be shorter, 3 to 6 months from amino acid sequence to R&D, to RCB. And the cell line stability is often less of a risk because of the nature of the details of the expression system. And so the 60 generation confirmation study may not be a critical path task, because it just is not as great a risk as it is with non-proprietary approaches. And then also very important to our client companies are the cost components. And you can see those indicated here. Proprietary systems generally involve fee-for-service and milestone payments, and not very often royalties. That's a variable, but it is something we need to look at as we evaluate a proprietary system. In contrast, the non-proprietary systems are public domain, and they produce a lower expected productivity, generally 1 to 3 grams per liter, with commercial media. Timeline is anywhere from 6 to 9 months through to RCB. 60 generation confirmation of stability is generally a critical path task. And so we want to make sure we do this, and we don't invest a lot of effort into parallel activities until we have a reasonable stability, a reasonable assurance that stability is there. Cost components are less of an issue here. Yes, fee for service, generally not milestone payments and not royalties. So going back to Tracy's point about time versus cost, generally, if cost is an issue, we'll steer a client toward a non-proprietary expression system, and that's kind of how this goes. Okay, so I mentioned that we would talk about special considerations for bispecifics, and I want to talk about that on this slide. So one special consideration is the design of the vector, and that's a single versus a multiple plasmid system. So as you know, bispecifics are more complex in structure. They require assembly of an oligomer that may vary with differential expression of the monomer chains. So in the example, the graphic that we have here, this is a bispecific that is full length in terms of heavy chains, but it involves 3 distinct polypeptide chains. There's a heavy chain on the left, it's an intact heavy chain. There's a hybrid chain on the right, and then a light chain. And so the chain ratio in this case, as with many bispecifics, needs to be optimized. And the best way to do that is to use multiple plasmids, which provide the greatest flexibility. One of the implications of bispecifics is that assembly is not assured. It's much more straightforward in a monoclonal assembly of the oligomer. And so we end up having a significant issue with variants of the product, which are inactive forms of the product that are assemblies of oligomers that are not the desired assembly. And so we try to minimize that both by vector design and in the expression system that we use, also in molecular design of the molecule itself. For example, knobs into holes technology that was first described in the 1990s at Genentech actually is one approach that's now off patent, so that's a public domain approach. There are several other approaches that direct the assembly of heterologous heavy chains to better assure the desired molecule is made. So those are important considerations in bispecific technology, bispecific expression, and these are considerations that are made at the very beginning of the program in terms of figuring out, how are we going to make this, and how do we design the expression system so that we minimize the formation of these variants. So structure and stability, we have bispecifics also have implications for the use of a platform process. Now, those of us in the monoclonal antibody world have become very comfortable with the platform process that we use for monoclonals. That platform process needs to be tempered and modified in order to make bispecifics. So for example, stability at extremes of pH is really important here. You need acid stability for capture on protein A and for low pH hold for virus inactivation. Well, bispecific antibodies are engineered molecules. Stability at extremes of pH are not guaranteed. And so that's something that needs to be evaluated early in order to determine how stable is this molecule at low pH. And if it turns out that it's not stable at low pH, then that has significant implications for the use of protein A as capture and ultimately on the platform process in general. The purification complexities that enter into the bispecific situation. As I said, the product variants, these are all considered impurities, and so they need to be removed. The challenges for removal of these in the downstream process are considerable, as any of you who have tried to do this will know. And so that's why we try to address as much of the problem in expression system and molecule design as possible, so that we reduce the burden on our downstream colleagues in trying to deal with this in purification. When we do have to deal with it in purification, the removal of variants falls largely to the final polishing step. That's the chromatography step, and that's going to be the major role of that final polishing step, is to deal with variants. So back to this case study. For this project, we went with Chinese hamster ovary cells and a proprietary expression system. In order to maximize the productivity of the cell culture process, the development focused on a number of factors. As you know, the titer of a process is determined by the accumulated cell mass times the cell-specific productivity, which is defined by the equation shown here, where QP is the cell-specific productivity and the integral of x dt is the cell mass. We multiply those together in order to get the titer. So what does a good process mean then in terms of maximizing these factors? A good process starts with a highly productive cell line. That is the QP factor. That's the cell-specific productivity. And a cell culture process that produces a high accumulated cell mass. That's the integral of x dt term. How do we get that? The time in production phase with high cell viability, we try to maximize this. The timing and composition of feeds, we want to provide just-in-time nutrients and minimize waste products. And the rate of agitation and oxygenation. All of these are the levers that we have to pull in a cell culture process in order to maximize the interval of XTT term. We can do this in a cell culture process design that's either fed batch, shown on the left, or perfusion, shown on the right. The more common approach in our industry today is fed batch. In the case study at hand, we did 3 2-liter confirmation batches and a 100-liter pilot batch. And so the productivities of these various batches are shown here on this slide, where we did 2 rounds of screening at 2-liter scale, where the 2-liter baseline had a titer of 2.3 grams per liter. That's shown on the left in the graph. And then through the first round screening, you can see that we were able to improve the titer up to 3.7 grams per liter. And then in the second round of screening, we were able to actually exceed 4 grams per liter. So when we then took the 2-liter process into a 50-fold scale up into the 100-liter tox lot, we were able to achieve a titer just shy of 4 grams per liter. We also changed the cell line from the research cell bank, which was all of the 2-liter work, to the master cell bank, which we used for the tox lot. Now, this is not required. Depending on the timeline, you can do the tox lot with the research cell bank and then switch to the master cell bank for the GMP batch. In this particular case, we had the master cell bank completed and characterized prior to the 100-liter tox lot, so we chose to do that batch with the MCB. So I talk now just about GMP production. Summarizing the scale up, we did the confirmation runs at 2-liter bench scale. As I said, we performed this with the research cell bank. And the confirmation runs at 2-liter scale are intended to confirm performance of the complete process. This is the opportunity that we have to combine the upstream with the downstream process and the formulation that has been developed. So it's taking all 3 of those components, upstream, downstream, and formulation, and put them together into a bench scale process at 2 liters. Tox production at 100-liter pilot scale, in this case, was performed with the master cell bank. And the pilot scale process reflects the GMP process. This is critically important for comparability of the tox production to the GMP production, both to justify the use of tox material for that purpose, to predict the toxicity of the clinical trial material, but also because the reference material is being generated from this batch, to which the GMP batch will be measured. The 2 processes need to be as similar as possible. GMP production at 2,000-liter scale was performed with MCB. Samples from this were taken for viral clearance study and drug substance and drug product stability. And also the assay methods for GMP production were qualified. In this slide, I'm showing you the confirmation runs. These are the 2-liter confirmation runs versus the tox batch. In terms of viable cell density, and you can see that over the 12-day time of the run, the growth of all 4 of these batches is comparable. Peak cell density about day 8, with a drop down to day 12 where we harvest. Viability in all 4 cases was also high. Final viability about 80% at day 12. Good place to harvest. You want to keep viability above about 70% for a biologics process. And the titer, as I said previously, for the tox batch was just under 4 grams per liter. Day 12, you can see that the titer measured from day 6 onward was pretty constant, pretty reproducible for all 4 of these batches. 3.9 grams per liter on day 12. So the summary of the process that was used here for this, in this case study, the upstream process is shown in blue, the downstream process in orange, leading to bulk drug substance. The fed batch process started with a vial of the master cell bank that was thawed into the seed train, which then became the expansion train used to inoculate the production bioreactor. In this case, that was at 2,000 liters for 12 days. We then harvested by a combination of centrifuge and filtration. And then the harvested clarified cell culture fluid went on to protein A. We were able to use multiple cycles per harvest. This is typically done with monoclonal antibody and bispecific processes to use multiple cycles. Why this is done is because, as many of you know, protein A media is one of the most expensive components of the manufacturing process. It is reproducible, and so over time that cost decreases. But this is clinical production, and in order to manage the cost of the protein A media, the best approach is to purchase a smaller amount of media and then cycle the harvest through the media several times, anywhere from 3 to 5 times. Cycling over protein A is typically done. That is then followed by low pH hold, which is a very effective viral inactivation step. The process here was followed by anion exchange chromatography. This is in a flow-through mode. And then cation exchange chromatography for final polishing to remove aggregates, or as I said previously, if this were a bispecific process, it would look similar, assuming that it's a full-length bispecific that binds to protein A, where the cation exchange column, which is in the final polishing role here, would be the column that is most effective at removing product variants. That's followed by virus filtration, typically 20 nanometer virus filtration. And this is a very effective virus removal step, followed by formulation by UFDF. We would want to see, and in this case, we did see 70 to 80% overall product yield going into the frozen bulk drug substance. For monoclonal antibodies, and bispecifics, as I said before, the drug substance and the drug product are typically the same formulation buffer. The compounding that's done here is very often the addition of detergent in the adjustment of product concentration. So formulation buffer would be introduced at UFDF. Detergent in the form of polysorbate 20 or 80 is added as a compounding step prior to freezing the bulk drug substance. The drug product process is shown here. The frozen bulk was thawed, filtered, then filled into vials, labeled and packaged, and the result of that is shown in this slide. Now, just to go back to what Tracy was talking about in terms of the CDMOs who participate in all of these activities, in our case here, this client was interested in one CDMO that would perform everything from cell bank vial here to through frozen bulk drug substance to filled into vials drug product. So we did all of that with one CDMO and then went to a second CDMO for labeling and packaging. That's what we call it. That's what we mean by one-stop shop, somebody who does both drug substance and drug product. Formulation development and drug product stability studies are outlined on this slide as they were performed. We developed a stable formulation for storage of the drug substance at -20 degrees C and a stable formulation for drug product for storage at 2 to 8. In order to do this, we evaluated pH and excipients, and then a formulation was selected based on 3-month stability data. Now this is risky, but it's what fits well into the timeline, and enough information can be had from a 3-month stability study that includes accelerated conditions and appropriate stability assays, so that it should work well. The goals here were a liquid drug product formulation at 10 mg per ml for IV administration, a stable drug product for greater than 24 months at 2 to 8 degrees storage, and a drug product that's compatible with a container closure system and in-use compatibility if the drug product is to be diluted in an IV bag, which it was in this case. As I mentioned, very important to these formulation development studies are key stability indicating assays. These must be identified and used for these kinds of studies. For monoclonal antibodies and bispecifics, one of the most important stability indicating assays is SEC, HPLC, because the principal route of degradation of these kinds of molecules is aggregation. So when did we time our interactions with FDA for this client? Well, they're shown here on this slide in blue with blue arrows. And so this is you saw this, this graphic earlier where we initiate cell line development at the beginning of the program. That's the white arrow. Our first interaction with FDA is the pre-IND meeting. We scheduled this meeting to occur after we produced the pilot material for GLP's tox studies, but before we actually initiated the GLP tox studies. And in this pre-IND meeting, we asked CMC and tox questions, and we provided the agency with preliminary drug substance and drug product stability. With the pre-IND meeting then completed, we did our GMP, our tox studies in our GMP manufacturer, and then we filed the IND once the GMP material was in hand with 1 month of drug product stability. So in summary, if you're a scientist at a company wishing to develop a new biologic for clinical testing, we suggest that you plan on outsourcing manufacturing to a competent CDMO. A CDMO can design a production process and can provide high quality product in order to meet the regulatory requirements that you'll face in sufficient quantity. Very importantly, as Tracy pointed out, CDMOs have particular expertise and their capabilities should be evaluated carefully. Plan on 1 to 2 years to develop a cell line and a process and to manufacture tox and clinical product. Plan your interactions with FDA around the uniqueness of your product. And you may want to seek a CMC consultant to assist in this effort. All right, with that, thank you very much for your attention, and we will be happy to entertain questions.
Terrific, Steve. Thank you very much. We have a couple questions that came in during your presentation today. The first one is from Lisa. And Lisa early on wanted to know if you'd encountered any pushback from the agency about doing IND enabling stability studies on BDS versus DP.
Yes. Yeah, Lisa, we have encountered that. The agency is pretty, and that, you know, I want to, first of all, I'm wondering if is this a general question or does this have to do with COVID? So I'll answer it as a general question. In general, yes, FDA wants to see stability for drug product because they're interested not only in the product itself, but they're interested in the product in the context of its container closure system. So I would say yes, we have received pushback from the agency on use of drug substance as a surrogate for drug product. That may that situation may be modified for COVID because of the priority there that's put on speed. We can talk a little bit about that if you'd like, but there are the agency has modified some of its decisions in order to facilitate speed for COVID, as many of you know.
Thank you. Our next question, Steve, comes in anonymously. And the question is, do you typically advise a one-stop shop or multiple CDMO? What would be the main reasons for multiple CDMO that would be chosen?
Specialized expertise. So I'm going to go back to the match that Tracy talked about. There isn't a typical. It all depends on what's the project, what are the complexities that we're going to face here, what are the priorities of the program in order to ensure success. So if we have a bispecific molecule and formulation is a big issue, or perhaps we're inheriting an expression system that is suboptimal and product variants are going to be a significant issue, then whether it's a one-stop shop or not is less important than getting a CDMO with the right technical expertise to deal with those issues. So we're going to want significant bispecific experience in formulation, development, and in purification. A one-stop shop is a nice to have. It's not a need to have. We can manage the performance and the delivery of clinical trial material from two CDMOs almost as well as we can from one CDMO. It's really not that big a deal.
Thank you. Our next question is from Vivek, and it's a multiple part question. So I'll read the whole thing for you. He says, do you advise a suitable platform for BSAB generation, full length IgG format? And what are the options to tackle mispairing and half antibodies issues in early stage of development?
Can you, Kelly, I didn't get that first part. Can you repeat the first part?
Yes. Do you advise a suitable platform for BSAB generation, full length IgG format?
So again, I'm going to go back to the initial discussion with the client. So that discussion is going to be what's your tolerance for proprietary versus public domain systems. There are several proprietary systems that are out there that are available for license that work very well. But if the sponsor doesn't want to go that route, then we're looking at public domain technology, and then we're probably talking about knobs into holes with and then having to deal with the issues that come with that. I mean, and it's a good system. It's not the best system, but it's a good system. We published an article a couple of years ago in Bioprocess International that reviewed, it was kind of an overview of all of many of the different technologies that are available for bispecific construction. And so I might refer you to that article to highlight, what some of the better systems are for bispecific expression. But yeah, that's one of the most important discussions to have at the beginning is what expression system should we use here, recognizing that the specific issues we're going to face with bispecifics relative to monoclonal antibodies.
And so then the second half of that question was, what are the options to tackle the mispairing and half antibodies issues in the early stage of development?
The most important way to deal with that is directed assembly, a knobs into holes or alternative approach, molecular engineering approach in order to direct assembly of heterologous chains. There are also approaches out there that use a common light chain to reduce the number of mispaired variants that are theoretically possible. That's worth looking at. At the end of the day, if you can, you do the best job you can in order to get as much of the product of interest in the crude mixture, and then the strategy is, or what do we have here, and can we exploit the characteristics of the product variants relative to the product of interest to achieve removal. So if PI differs, for example, between the product variants and the product of interest, then cation exchange or mixed mode chromatography can work well in that polishing step. If there are hydrophobic differences between the product variants and the product of interest, then something like Capto-Q or hydrophobic interaction chromatography can be exploited in that same role. That's how I would approach that.
All right, well, those are all the questions that we have for the webinar today.
Okay, well, very good. Again, I want to thank you all for spending the last hour with us. If you have any further questions, please feel free to contact Tracy or myself. And thank you very much and have a good day.
Thank you.